Hi, everybody. Thanks for tuning in today, and good evening or good morning from wherever you're coming from today. My name is Kym Ottewell. I'm a fauna geneticist working in the Department of Biodiversity, Conservation and Attractions in Western Australia. Firstly, I'd like to thank the conference organisers for the opportunity to talk today on how we're integrating genomic data into applied conservation management. And I'll be using some examples from threatened species that we're working on here in Western Australia. Before I begin, I'd also like to acknowledge that I'm presenting today from the traditional lands of the Whadjuk Noongar people, and I'd like to pay my respects to them, their culture, and to their elders, past and present. So most people will be keenly aware that we are in the midst of a global biodiversity crisis. Australia, in particular, has had an unenviable record on species extinctions, especially of our mammal fauna, with nearly 1/3 of recent global mammal extinctions having occurred in Australia over the last 200 years. Threats to our wildlife are pervasive and stem from extensive land use change, introduced feral predators such as the European fox and the cat, inappropriate fire regimes, whether this is too frequent or infrequent fire, which has been leading to ecosystem change. And as a result of this, many species are experiencing massive range collapse. And despite our collective efforts, threatened species populations continue to decline, which is eroding our natural capital. So while predators and habitat loss are some of the extrinsic threats to wildlife that are quite well understood, perhaps one of the more insidious threats that is currently under-recognised is the ongoing loss of genetic diversity. So genetic diversity underpins biological diversity and ecosystem functioning, so it contributes to species and ecosystem resilience in the face of environmental change. So for species that have undergone rapid population declines, while we may be able to intervene and use conservation interventions to improve the demographic recovery of those populations, once we lose genetic diversity, that's lost until new mutations arise, which is a very slow, ongoing process. So these has been highlighted recently in this article in Science that indicates that we may be already exceeding some of the targets we've set within the UN on halting genetic diversity loss. So the Department of Biodiversity, Conservation and Attractions is the state government agency in Western Australia. Western Australia covers about 1/3 of Australia's landmass. So as a conservation agency, we're actually responsible for quite a large proportion of management of threatened species in Australia, managing over 25 million hectares of conservation estate, 429 threatened flora, 249 threatened fauna, and 65 threatened ecological communities. So this is not such a small task. One of the major conservation management actions that we undertake with threatened species in Western Australia is the use of conservation translocations that are used to improve the conservation status of threatened species by re-establishing populations within their former range or establishing new populations in terms of conservation introductions into refuges outside the species' former range. And we also undertake augmentations to existing populations. Typically, a secondary goal in many of our translocations is to restore ecological function into those degraded habitats by replacing some of those functional traits. So DBCA has been particularly active in this area. We've undertaken more than 230 translocations of 43 species to over 142 sites over the last 30 or so years. And due to the sensitivity of translocation process to loss of genetic diversity, we are considering genetic diversity explicitly within each step of the translocation process. So this tends to begin with our pre-translocation planning, where we use genomic data to survey and identify suitable populations for translocations. So here we're considering genetic diversity, inbreeding status of populations, as well as their divergence history. And frequently we are using genetically informed population viability analysis, or PVA, to evaluate different sourcing strategies as part of our planning. So, for example, this might involve assessing the use of single versus multiple sources in translocations. We consider the harvesting impacts on source populations. We use PVA to model appropriate founder group sizes needed to establish those genetically robust and representative populations. And in the longer term, we want to ensure that populations are self-sustaining and able to conserve genetic diversity through time. So today I'm going to be talking about a few examples on how we're integrating genomic data into our conservation practices, usually using the Dirk Hartog Island National Park Ecological Restoration Project as examples. So this project is an ambitious project, and it's one of the largest fauna reconstruction projects in the Southern Hemisphere. So just to set the scene, Dirk Hartog Island is located on the western coast of the state of Western Australia, and it's located in the Shark Bay World Heritage Area. The island is known as Wirrawana in the local Manguna language, and it's an island of approximately 70 kilometres stretching from north to south. It's famous for two reasons. Firstly, it is the westernmost point of Australia, and it is also the site of the first recorded European landing in Australia when Dutch merchant and explorer Dirk Hartog visited the island in 1616. And to record this event, a pewter plate was nailed to a wooden post towards the northern end of the island. So Dirk Hartog actually operated as a pastoral lease, as a sheep station for about 150 years. In 2009, Department of Biodiversity Conservation and Attractions were in negotiations to return the island to National Park and to undertake ecological restoration, and that was gazetted in 2009. So the aim of the Ecological Restoration Project on Dirk Hartog Island was to restore the island to its original condition that the explorer Dirk Hartog would have experienced when he came ashore in 1616. And this is involving the reintroduction of 12 mammal species and 1 bird species back onto the island. So the ecological restoration component of the project began shortly after 2009 and involved removing sheep and goats from the island. The feral goats from the island were eradicated, and then concurrently work was undertaken to remove feral cats from the island. And following 2 years of no sightings of cats, the island was declared cat-free in 2018. And so this began the fauna restoration component of the project, and this involves reintroducing some of Australia's most threatened mammal species. So translocations are now well underway. We translocated banded and rufous hare-wallabies from nearby Bernier and Dorrie Island onto Dirk Hartog Island in 2018-2019. Since then, we've released the Shark Bay bandicoot and dibblers in 2019 and 2020, with some further top-ups for the dibbler in 2021 and 2022. And last year we reintroduced both greater stick-nest rat and the Shark Bay mouse onto the island. The translocations of the western grass wren are coming up as we speak, really. And in 2023, we'll be translocating Mulgara onto the island, and I've just completed the genetic analysis for that species. The remaining species, we have woylies, booties, chuditch, desert mouse, and heath mouse, and these will be released onto the island over the next coming years. So also, although the booty, the burrowing bettong, is actually later on the list for translocations, it actually serves as a really great case study on how we're using genomic information to benchmark genetic diversity across species and to identify genetic restoration targets. We've learned a lot from previous translocations of this species, and we're now incorporating this information into our current translocation practices. So the booty, as one of only 2 fossorial marsupial species in Australia, is actually regarded as an ecosystem engineer. The species lives in communal burrows, and their burrowing activity contributes to soil turnover and nutrient cycling in these arid landscapes. Burrows also provide suitable microhabitat and shelter for a whole range of other species and provide protection from predators. So a very important component of the ecosystem. Prior to European colonisations, booties were actually widely distributed across much of Western and Central Australia. But with advancing agricultural practices and the introduction of feral predators, the species has undergone a massive range collapse and is now only found in 3 remnant island populations on several islands off of the coast of Western Australia. So 2 of these islands, Bernier Island and Dorry Island, are located in the Shark Bay World Heritage Area and adjacent to Dirk Hartog Island National Park. And a 3rd population is located on Barrow Island, which is a bit further north of the Pilbara coast, and is also recognised as a distinct subspecies compared to the Shark Bay Island subspecies. So the booty has now actually had quite a few translocations over the years. It's been successfully established in a number of alternative island populations, and as well as a number of fenced reserves on the mainland. So as part of excuse me, as part of our translocation planning for Dirk Hartog Island, our plan was to evaluate the genetic diversity across remnant populations, as well as these reintroduced populations to determine appropriate source populations for translocation. And as you can see with these reintroduced populations on the mainland, the colours of the circles actually represent the source populations used in these reintroductions. So we've actually got different combinations of populations being used in those reintroductions. So it's a good opportunity for us to investigate the impacts of genetic mixing in this species. So this work was actually undertaken as part of the OzMammal Genomics Initiative that provided sequencing resources for the project, and in collaboration with a whole suite of collaborators from NGOs, universities and museums, so who provided us samples both from these reintroduced populations and also historical specimens that we were able to obtain. So for the contemporary samples, we were able to use ddRAD and to obtain population genomic sequencing data for these populations. And as part of the OzMammal Genomics project, the development of the marsupial, the universal marsupial exon bait capture kit was something we were able to use to obtain genetic data from museum skins, as well as from a subset of the contemporary populations. So these museum skins, we obtained 7 specimens from the southwest of WA in particular, and these represent samples that were collected between the years of 1896 and 1928. And further, just because of the special interest in the Madawa population, which is the only population where the Barrow Island booty was mixed with the Shark Bay Island booties, we actually have extensive temporal sampling from this population in the ddRAD data, which has allowed us to gain more insight into the patterns of admixture and integration in this population. So as I mentioned, one part of our pre-translocation planning is to identify the suitable genetic restoration targets for the booty. So here we benchmarked genetic diversity across the remnant island populations and compared that genetic diversity to data obtained from the museum specimens, which we used to represent the historical, now extinct mainland population of booties. So SNP data was extracted from the exon capture reads, and levels of individual heterozygosity was estimated across those populations. And this work was undertaken by ANU postdoc Emily Roycroft, who's a collaborator on this project. So our first results indicated that the mainland population, even though this was only a sample of 7 specimens and really from quite a geographically restricted corner of southwest WA, still retains significant genetic diversity. And we can see that that genetic diversity from the mainland has been lost from these island remnant populations. So these islands have been isolated from the mainland in the case of Barrow Island for about 10,000 years. And in the case of the Sharp Bay Islands from Bernier and Dorey Island, they've been isolated from the mainland for about 6,000 years. And these populations are relatively small and have undergone ongoing bottlenecks. So really, so this result, I guess, isn't a terrible, surprising result, as we did anticipate experiencing low genetic diversity in these populations. But having the mainland samples from the historical specimens, this actually gives us a genetic restoration target to work towards for the species. So our second objective was to identify the optimal translocation strategies for the booty. So given that the island sources really were insufficient on their own to contribute to genetic diversity restoration, we were actually interested in seeing whether we can learn anything from the previous translocations across this species and how differing sourcing strategies may be used to improve genetic diversity. So in this part of the project, we were benchmarking genetic diversity across all of the contemporary populations, which was the remnant populations, single source populations, as well as the mixed source reintroductions. And this was done using the DD-RAD data run by postdoc Heidi Nistelberger, and this is the data set where we had exhaustive sampling across all of those contemporary populations. So the DD-RAD data was aligned to the closely related Woylie reference genome, and from this we estimated population structure and obtained standardized standardized heterozygosity estimates. To put the genetic mixing question into context, I guess, we needed to understand population structure amongst those remnant populations of booties on the islands. So the DD-RAD data showed us very clearly that each of the island populations is genetically distinct. Barrow Island is slightly more differentiated from the Sharp Bay Islands, but the Sharp Bay Island populations, Bernier Island and Dorey Island, are also significantly differentiated from each other. And this is likely, again, due to that degree of isolation between the islands that have been isolated from each other for about 3,000 years once the land bridge was eroded between them. So FST results are very high, not unexpectedly, I guess, across these island populations. And this really reflects the patterns of genetic drift that are occurring in these islands. So in terms of the genetic mixing question, we're looking at the impacts of mixing the Barrow Island and the Sharp Bay Island in the Matterhorn population, the reintroduced Matterhorn population, compared to using the two Sharp Bay Island sources in these other reintroduced populations run by AWC and Arid Recovery. Here are our standardized heterozygosity results that look at the impact of mixing on genetic diversity. So quite clearly, first of all, you can see that the remnant island populations and using single source translocations has just minimal genetic diversity and very similar genetic diversity across all of those populations. However, when we mix, excuse me, when we mix the divergent source populations in Matterhorn, we mix the Barrow Island and the Dorey Island source populations together here, we get a big boost in genetic diversity. This is significantly different to any of the single source populations. And when we're mixing, even when we're mixing the two divergent Sharp Bay Island populations together, that's Bernier and Dorey Island, we do again get that the bump in genetic diversity there. So switching back to the exon data, exon capture data, where we do have that mainland target, does mixing actually get us anywhere towards our genetic restoration target? So we can see mixing within subspecies gets us somewhere there, but mixing between the divergent, the more divergent Barrow Island and Dorry Island sources in the Mattawa reintroduction, that gets us closer to our mainland target. Because we only mixed the two populations in the Mattawa population, we can imagine if we did mix all of those three island populations that were still very diverged from each other, we might actually recapture some of that those subsets of genetic diversity that were present in the mainland population. So mixing divergent populations, I guess, is not without risk, however, and has been quite a controversial idea in conservation biology, mainly due to concerns over outbreeding depression, which is the loss of fitness that may occur when two divergent populations are crossed. Fitness loss may be a result of intrinsic genetic incompatibilities between the two mixed populations, or it may also represent lowered fitness that occurs as a result of the loss of local adaptation, in which those new mixed populations may be maladapted. So due to these theoretical concerns, there really has been hesitancy to mix populations in threatened species management, and this tends to result in a decision to stick with the status quo and leave populations as is. So this also has its own set of consequences in terms of managing the ongoing loss of genetic diversity and inbreeding in small populations. So there is now beginning to emerge some awesome empirical data showing the impacts of genetic rescue or evolutionary rescue in some cases, where mixing differentiated populations can actually lead to an increase in genetic fitness of the resulting population by reducing inbreeding depression and reducing genetic load. So the classic example of this work was done by Andrew Weeks with the mountain pygmy possum, and he and his team showed that the introduction of a small number of males into this declining mountain pygmy possum population actually resulted in a significant and rapid increase in population growth. So their data showed that genetic diversity was increased in this population as a result of those translocations. And in terms of fitness, those female hybrids were shown to have a longer lifespan and produce more young, which contributed to that population increase as a result. So we've already seen that the two subspecies of booties that we're working with are highly genetically differentiated, but they are also significantly differentiated in morphological characteristics as well. And the most obvious of these is a difference in their physical size. So the Chappé Island booties are actually almost twice the size as the Barrow Island booties. So given this disparity in terms of both the morphological differences and the genetic differences between these populations, we were concerned that mixing of these populations may have been a candidate for the expression of outbreeding depression. So the Mattawa population, where we mixed Barrow Island booties with animals sourced from a Dryandra population, which was originally sourced from Dorry Island, is one where we can actually investigate this question in a lot more detail. And this was the subject of honours student Kate Rick, who completed her thesis in 2019, where we were able to use comprehensive tissue sampling across founders and annual cohorts of booties that allowed us to investigate those patterns of expression of admixture and fitness. We had annual trapping results for this reintroduction as well, so that enabled us to actually use that data for microcapture analysis to estimate fitness impacts. So the Madawa population was established from 86 individuals in 2010 or 2009 and 2010, and grew to a population of about 750 individuals by 2018. So using this this data set, Kate was able to find evidence of interbreeding between the two divergent source populations in those first few years of the translocation. So that was evident here with the F1 hybrids detected as a half-half mix of those two different source populations. And then over time, she detected increasing admixture in those populations, which led to an increase in observation of F2 and back-crossed hybrid individuals. So just on the surface, this indicates that interbreeding between the two different subpopulations has actually been successful, and that latter generation hybrids are also being formed in this population. So using the genomic admixture proportions, Kate was able to classify her individuals into their different hybrid classes. And then with the comprehensive monitoring data that we have available for these specific individuals, she was able to use mark recapture analysis to investigate their patterns of survivorship and reproductive fitness. So firstly, the general observation we found was that because this reintroduction was into a fenced predator-free reserve, we noticed that survivorship is actually quite high across all of our hybrid cohorts. And we didn't detect a significant difference between males and females in terms of survivorship in these population. So we, as a baseline to establish sort of fitness effects in the Madawa populations, we found that the Barrow Island founder individuals and their offspring, as well as their Dryandra founders and their offspring, had very similar levels of survivorship. So there was no survivorship bias either way with those two original source populations. With the increasing hybridization, genetic theory would suggest that we are likely to see that fitness increase in the F1 generation, particularly as a result of heterosis or hybrid vigor. And the theory suggests that outbreeding depression is most likely to be observed in the F2 generation when admixing is actually breaking down those locally adapted gene complexes, which is leading to maladaptation. So our results for the booty population at Madawa were consistent with this increase in fitness in the F1 generation, but contrary to expectations under a model of outbreeding depression, that fitness effect was actually retained in the F2 generation, or at least we did not see a significant decline in fitness in that F2 generation that we might have expected to see if outbreeding depression was present in the population. So this was very positive news, and that was a very successful outcome for us, even though the populations had been significantly genetically diverged. To have no fitness impact upon mixing, that gave us confidence to sort of proceed. And in terms of genetic diversity, we also detected, we can also explore that pattern of increase in genetic diversity over time in that population. So we see a rapid increase in both allelic richness and heterozygosity in the Madawa population, and that this is enduring across multiple years in this translocation. So we have been very fortunate to have received good support through the Bioplatforms Australia genomic sequencing initiatives that are run here. So both the Mammal Genomics and the more recent Threatened Species Initiative has supported genomic sequencing for many of the species included for reintroduction to Dirk Hartog Island. And we also use a commercial service on occasion that's diversity arrays technology to provide that genomic data. So we will have foundational genomic data for each of the threatened species that are being released onto Dirk Hartog Island. And as this planning process, we're actually surveying most of these species sort of across their distributions. So the knowledge that we're gaining out of this genetic data is actually being used to support translocations of those threatened species in other areas of Australia as well, not just Dirk Hartog Island. So those investments under those sequencing initiatives are really paying dividends in terms of threatened species conservation. So now I wanted to shift a little and talking about how we are using genomics in an applied context in terms of genetic monitoring. As mentioned at the start of the talk, we embed genetic success criteria into our translocation practices, which means that we must undertake genetic monitoring on these populations to ensure that those success criteria are met. So there's been quite a sustained push, I guess, in the literature recently to embed genetic diversity targets into our global conservation frameworks. So this has been obviously very admirable, and it's something that we are actually getting more and more involved in. So I think at the local scale, we have conservation agencies that are very invested and interested in doing this type of work, but there's still a bit of a limitation in terms of the funding available to do this, and also the genetic methods to being able to achieve this sort of cost effectively. So in the Dirk Hartog Island project, I did mention we have genetic targets written into our post-translocation success criteria. So namely, these are ensuring that populations retain 95% of the genetic diversity of our founder groups. And they also exist as triggers for additional conservation intervention if these criteria are not met. So in being able to routinely undertake genetic monitoring for so many species, particularly on Dirk Hartog Island and in perpetuity, we really need genetic solutions that are actually affordable, and preferentially some that are low tech, so that our regional conservation staff can undertake the routine monitoring component of this translocation. And we also need methods and methodologies that are pretty much future-proof. For genetic monitoring, we want technologies to be stable so that we're consistently monitoring the same genetic markers over time, and we cannot really be affording to switch as new methodologies come on board, which is something that is happening very rapidly in the genomics space. So for many species where some existing genomic data is already available, and preferentially at the population level, we are actually making good use of the automated targeted SNP genotyping panels that assist us in actually meeting these criteria in terms of a genetic monitoring methodology. And I just wanted to go quickly through one of the somewhat bespoke approaches that we are developing for some of the species on Dirk Hartog Island. So banded and rufous hare-wallabies were two of the species that were first translocated onto Dirk Hartog Island in 2018 from remnant populations on the neighbouring Bernier and Dorey Island. So both of these species have proved incredibly challenging to monitor. Both of the species are highly cryptic, and particularly in the dense vegetation present on Dirk Hartog Island, it makes it very difficult to actually capture these animals, or to observe these animals and to capture them. Neither of the species readily enter traps, and both species, unfortunately, are actually quite susceptible to capture myopathy during capture and handling. So for these species, we've really had to focus on developing non-invasive monitoring survey methods. Non-invasive methods also give us an opportunity to provide more cost-effective monitoring rather than running full trapping or cam and trap survey programs on the island. So initially, we investigated the use of fecal DNA analysis as a methodology for genetic monitoring, firstly in the banded hare-wallaby, which was also extended to the rufous hare-wallaby. So we initially trialled microsatellite markers to achieve this, and in one of our early papers, we were testing the stability of DNA on scats exposed under different environmental conditions on Dirk Hartog Island. So we found that the methodology was successful. We were actually able to obtain DNA from scats quite readily, but that the exposure to different conditions, environmental conditions, really impacted on the DNA quality and quality of genotyping obtained from those scats. So for scats exposed to sun, we were only able to obtain confident in genotypes obtained for scats less than a week old, whereas those that were located in shade, we were still able to obtain genotypes from scats up to sort of 2 weeks to 3 weeks there, which was quite pleasing to know that we have a bit of flexibility in obtaining DNA from these samples. So we actually ran a pilot study to genotype scats across a large transect plot on Dirk Hartog Island. And so we were able to, because we did have species-specific genetic markers, at this point in time, we were actually able to distinguish the two species from the scats from the two species from each other, which was very useful. And also I thought quite interesting that it really picked up their differences in the way the two species are using the habitats, and pretty much they do not interact. They do not go near each other. So banded hare-wallabies tended to be in the vegetation towards the edge of the plot, whereas the rufous hare-wallabies really dominated in that lower vegetation through the centre of the plot. So at this point, we were interested in undertaking microcapture analysis using the microsatellite markers to identify individuals within this plot. And while we did have some success in identifying individuals, the markers were impacted by allelic dropout and also low variability between individuals, which sort of really limited our confidence in being able to confidently undertake microcapture analysis to get a population estimate. So following on from work that I'd done with another species, the ghost bat, that I'd been developing targeted SNP panels for, we actually decided to focus on developing a targeted SNP genotyping approach for both banded and rufous hare-wallabies. So fortunately for both species, as part of the OMG project, we had RADseq data available to us, and this was at the population level, so we did have good information on characterising those SNPs in each of the source populations. And really with targeted SNP genotyping, SNP selection can be infinitely flexible. You can design your targeted SNP panels to get you information on whatever you'd like, really. So we focused on identifying SNPs because we were interested in individual identification as a microcapture technique. We focused on identifying SNPs that had high information content. So these are SNPs that had high minor allele frequencies and also high levels of heterozygosity to provide that ability to genetically discriminate between individuals. We also, because the two different island source populations were used in this translocation to Dirk Hartog, we were also interested in detecting patterns of integration or demonstrating successful interbreeding between those two source populations in the Dirk Hartog Island translocation. So we developed island specific markers to include in the panel. So we use the mass array platform, which involves designing allele-specific primers from the short sequencing reads, and then multiplexing these to allow for automated genotyping on the mass array platform. Once we received the genotypes back from AGRF, who run the mass array platform, we undertake matching of genotypes amongst scats to be able to identify individuals, and using those island-specific markers, identify those hybrid individuals within the new population. We have a custom-made R package available to do those sorts of analyses. And once we have information on individuals, we can then use that in our monitoring approaches to estimate population sizes using microcapture information. The data give us information on spatial distribution of individuals, so we can look at home ranges, dispersal, patterns of integration. We can also monitor genetic diversity in the populations, as well as get information on relatedness within the population as that progresses through time. So at this stage, I only have some trial results to present today, but I did want to sort of just walk through some of the features that were built into the SNP panel design. So as mentioned, we focused on identifying markers that were available to provide high discrimination to identify individuals. So we used high minor allele frequency SNPs with a heterozygosity range between 0.2 and 0.6, and this gave us a probability of identity of using 18 markers to have high confidence in discriminating between related individuals. Our following analysis, we can show here in the heat map that we actually get really good discrimination between individuals. So we ran a trial of both tissue samples and scats with high success rates obtained for the scats, and along this horizontal axis here, we show those dark colours are individuals. We do find in some of the scats we were actually able to group multiple scats from single individuals together. So we do have that ability to group scats by their individual IDs. As mentioned, we also were interested in patterns of introgression in the Dirk Hartog Island population. So our initial trial, we genotyped several individuals that were known to be from Dorey Island and from Bernier Island, and we had a bunch of scat samples we had unknown identities for. So this analysis, we were actually able to run a structure analysis using the island-specific markers. These are markers that are fixed at alternate alleles in each of the populations. So we're able to identify purebred Dorey Island individuals, purebred Bernier Island individuals, and here we were able to detect several hybrid individuals. Mostly, I would say we're looking at F1 hybrids, but we might have a case here where this might be one of the back-crossed individuals. So this allows us to sort of summarize this information to determine the patterns of introgression in that new and establishing population. The SNP panel is also going to be used to estimate population sizes through the microcapture analysis. It gives us estimates of genetic diversity across the populations. We also have sex-linked markers not included in the panel that we run as a separate assay, so we can incorporate that information to look at sex-specific patterns in that population. And given that the markers are designed to have a high information content, this gives us the ability to estimate pedigree relationships within those populations. So as a genetic monitoring tool, I think targeted SNP genotyping really fits the bill in terms of giving us an affordable, cost-effective, reproducible approach to genetic monitoring. So the SNP panels really have a highly flexible design. So here we are focused on sort of anonymous markers, but you may also be interested in monitoring diversity at, say, some functional gene, which you would have the opportunity to include SNP markers in the panel to investigate these. We find that the mass array platform that we're using in particular has been suitable for non-invasive samples, so for scat samples, and we're about to test hair samples. The setup and running costs are affordable for these approaches. So the mass array in particular took, this is in Australian dollars, about a 3,000 K setup, whereas running each sample is, the costs are down around $10 to $11. As the genotyping is automated, there is minimal processing to be done, both in terms of getting the samples to the sequencing provider, but also in that SNP genotype data that is returned to us. The pre-selected SNPs mean that we know that they are high quality SNPs, and we quite often have really high quality data coming back from these platforms. The SNP panels are standardized, reproducible, which makes reporting over time a lot more streamlined and comprehensive. But I would say that one of the main limitations of this platform, in particular the mass array platform, is that it only is designed for a small number of markers. So each of our SNP panels contain about 50 markers, so which I guess doesn't give terribly good information, I guess, on genome-wide genetic diversity metrics, but at least it does give us a reproducible method starting off with this type of genetic monitoring. So I guess I did have a question for the audience. If anyone else is actually undertaking these genetic monitoring approaches, what platforms are people using, and what are the pros and cons of the different methods that people have been using? I'd be interested in gathering more information on different approaches. So I guess my ideal dream for these SNP panels is to develop SNP panels or similar appropriate design for a range of threatened species, and to have this available at the national level so that it is something that could be applied to different populations, and we can actually achieve comprehensive genetic monitoring across species distributions and in translocated populations. And this, I've just done a mock-up of, say, a scorecard type report that I can see this type of information being used in our ongoing reporting in terms of conservation management of threatened species, and integrating this into workflows for conservation managers. So I think I'll just end up there, and would quickly like to say a big thank you to all of the collaborators that have been involved in these projects, particularly to the staff as part of the Dirk Hartog Island Restoration Project. Sol Cohen and Colleen Sims have been running that program. We've been incredibly grateful to have had sequencing support from the Bio Platforms initiatives, which has really given us that foundational information that we need for our threatened species. And we've developed some awesome collaborations with researchers at a bunch of universities and other NGOs and museums. And of course, Pawsey, who provide the supercomputing facilities to undertake analyses. So I will leave it there. Thank you.